In this video, we will discuss about how this RBPJ regulates FOXP3 in activated T cells. In our previous video, we have discussed that effect of RBPJ on FOXP3 that it has a negative effect. If RBPJ is high, then FOXP3 is low. So now, in order to validate how this RBPJ affect this FOXP3, what it is, we use the high mutagenesis in this RBPJ and then check which domain will affect this FOXP3 expressions or enrichment, and they are able to find that this beta trefoil DNA binding and CTD domains cause high FOXP3 expressions. The mutations in this cause high FOXP3 expressions. When they check the beta trefoil DNA binding domain and CTD, it mainly interacts with the complex SMR and core complex. So they think that RBPJs forming a complex with this SMR and core plays a role towards this FOXP3 upregulations or downregulations. So first they check the effect of the mutant RBPJ. So in one case what it is, this mock control where RBPJs are present, and then in one case the RBPJ is overexpressed where we can see downregulation of the FOXP3, and in the third case where RBPJ is mutated at two site, that is at 225P, 235F, 235 mutated with A, and L362 mutated with A, and these two mutations lies in the beta trefoil DNA binding domain and C-terminal domain, and they can see that it helps in the recovery of the FOXP3 expressions. Thereafter, they wanted to see how this RBPJ, whether the RBPJ directly interact with the FOXP3 during the transcription process or not. So what it is, they want to check the FOXP3 starting transcription promoter sequence, and then they introduced the promoter sequence with the luciferase, and in the wild type conditions what they are able to see, where they are able to see very low signals because RBPJ is present, and when the RBPJ is been knocked down, they are able to see increased signals of the FOXP3, and when the RBPJ is mutated, they can see further increased signals in the FOXP3, indicating that the RBPJ directly interacts with this promoter sequence, starting sequence TT, CCAC, and when it is mutated, the interactions of the RBPJ with FOXP3 does not occur, and that leads to the high luciferase signals, since its translations will not be affected by the interaction with the RBPJ. And thereafter, and here is the relative luminescence or luciferase units, where we can see that in the mock luciferase signals are high, whereas in the case where RBPJ has been overexpressed, the luciferase signals are lower. So here it shows about the luciferase effect when the RBPJ is overexpressed. Now they wanted to check the interactions of the RBPJ with this SMR and core. So, or we can say this complex is HDAC3. So in the conditions where RBPJ is overexpressed, they immunoprecipitate the RBPJ and they check the presence of the HDAC3. They are able to see HDAC3 in the where RBPJ is overexpressed. However, when the RBPJ is been knocked down, they do not see HDAC3, indicating that this HDAC3 does not form a HDAC3 is form a complex with the RBPJ, but in the mutant conditions is not able to interact with the RPAG. So next they wanted to check, they see the deacetylizations of the FOXP3, and they can see that in the case where RBPJ is knocked down, the more deacetylizations in the activated T cells compared to the control, and deacetylizations mainly at H3AC and H3K9Cs they are able to see these two deacetylizations are more in the case where RBPJ RBPJ has been knocked down, and when the RBPJ is overexpressed, I forgot to explain this panel before going to the panel. In panel, when the RBPJ is overexpressed, they can see that uh, in the control case, the FOXP3 is low. However, when the HDAC has been knocked down, the FOXP3 overexpression is recovered and it is high. However, in the mutant conditions, they can see no change in the control and in the HDKC knockout, indicating this is the HDAC knockout. HDAC, RPPJ complex plays a role towards this FOXP3 formations, and the quantifications is shown in the bottom side of the panel E, where in the mock case they do not see significant changes, but when the RPPJ is been overexpressed, they see changes in the FOXP3 expressions. That is, when the HDAC is knocked down, the FOXP3 expression goes up, and when the RPPJ mutant is used, they do not see any significant FOXP3 expressions either in the control condition or HDAC is knockdown conditions. This indicates the complex formations of the RPPJ with HDAC3 plays an important role for the FOXP3 regulations. Now through this their technique, that is in ATAC and this in-chip seq, or it is written that cut and run, they wanted to optimize this deacetylizations that we have discussed, and in the H3AC and H3K9AC, and they can see that in the case of this RPPG overexpressed and RPPG mutant conditions, they are able to optimize using the in-chip seq conditions. The expressions of the H3AC and H3K9ACs are been optimized so that this FOXP3 expressions does not get affected too much in the case of this RPPJ is been overexpressed, and they made it comparables to the and the RPPG expressions are made comparables to the case where RPPG mutant is used. So this also makes sure that FOXP3 expressions does not get over affected. So this is how they use this deacetylizations results to modulate this H3AC and H39AC, that will help in the stability of the FOXP3 formations. So, and this is how they provided mechanisms of how this RPPG regulates the FOXP3 in the activated T cells. If you need to understand more about their experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.